Okay, so welcome to the Gerontomics workshop. This is a project that we have been carrying out for the last 5 years. It is funded by an operational competitivity program which ran from 2014 to 2020, and it was extended for one additional year. And it was funded from the European Fund for Regional Development. The project was aiming at understanding aging by developing tools for multi-omics analysis and prioritization of gerontological interventions. Actually, one question is if you can see the whole screen, because I see also the participants. So I hope you do. In aging research, the focus is on why people age. This is a trivial question if you ask random people, because intuitively we do understand what aging is. You have young people who are very active and developing. They are growing. As you can see, there is a picture of me when I was young and probably very foolish. And then with time, people change. We become more serious, probably. We start talking about grants and research. And then based on some simulation, probably this is how I will look, being less active and with continually decreasing functionality. However, if we start talking about aging at a molecular level, things get a bit more complicated. So one of the main things that you would see is that with age there is an increased probability to die, and depending on the species, there are different curves in which the survival decreases. So you can see here on the right some examples. Our species, because it's a protected species, protected by the society, has a relatively long average lifespan, and then at the end of the life, things get worse in an abrupt manner. Molecularly, there are a lot of things that change with age. There is genomic instability, telomeres get shorter, then there are a lot of alterations at the epigenetic or mitochondrial level. Nutrient sensing is deregulated. Proteostasis is not as it used to be. With time, you accumulate cells that are not functional, and this is cellular senescence. Stem cells cannot replenish everything as they used when you are young. Intercommunication between cells is going haywire, and so on. So there are many things, and the picture becomes more and more complicated. So probably that's why solutions to this problem are not trivial. So two of the main questions that or on the minds of researchers are: why are species aging differently? Why do they have different lifespans? For example, you have species that live a few days, like insects or some simple organisms, and then you have more complex organisms that live up to even 200 years, like whales. Of course, there is a lot in terms of genomic differences, but even among closely related species like mice and rats and naked mole rats and maybe bats, there are huge differences. So mice, for example, are short-lived animals, while naked mole rats are long-lived animals. Their size, which is generally the main determinant of longevity, is not that different to explain all this difference. So there must be something molecularly that triggers aging. The second question is what drives aging within a species? And we know there are genetic components and there are also environmental components. We know this because there are some progeroid syndromes, very nasty diseases, which make people look older much earlier than they should. We know this also because a lot of experiments have been done on model organisms, and a lot of genes, more than 2,000 genes, have been shown to influence lifespan. And we also know that some environmental changes, like caloric restriction, for example, can modulate aging. This means that probably contrary to the general take of the population, aging is something that we can work on and is something that can be somewhat effective. So besides the two main questions that fundamental researchers are working on, there are in general two practical directions that gerontologists focus on. One is predicting biological age. This is important because when you can estimate the age of an organism, then you can much easier evaluate if an intervention or a change in lifestyle can have a beneficial or detrimental effect on lifespan. The second direction is simply trying either blindly or informed to do interventions, for example, genetic interventions, and see if the organism lives longer or shorter. Now, this has been done in the past so in the beginning it was done with various information on what the genes could do. Then at some point, even in genome-wide screens. And we know now that there are a lot of genes that determine or have a role in determining lifespan of a certain organism. Most of these genes are discovered in mice, flies, worms, and yeast. And I think in C. elegans, there has been a lot of work. Probably most of the genes have been tested in a single genetic intervention pattern, let's say, or not pattern, but manner. Some of these genes extend lifespan, some of these genes reduce it, and also depending on the intervention that you do, they can be cataloged as pro-longevity or anti-longevity, because the interventions that you do are also either overexpression of the gene, increasing its function, or knockdown, knockout, or deletion. This is a summary from the GeneAge database. As you can see, there are genes that extend lifespan by even tenfold in worms. These are extraordinary cases, not too many. And you even have genes that can extend lifespan in mice, which is probably much more relevant for us humans. The main problem moving forward, now that we have a lot of information on the effect of genes on lifespan, is how do we combine this genetic intervention? And this is a even much more complex problem than before, when they had to, let's say, at maximum, spend a lot of money for genome-wide screen, because the number of combinations of genes grows exponentially. And another issue here is that probably because the problem is so complicated and so costly, researchers are less inclined to test interventions in multiple genes. And we will have a nice talk about this in the first session. Gabi will probably tell you a bit more about Synergy Age. This is a project that we did to actually evaluate the existing knowledge in the field on this. So the aim of the project that we received funding 5 years ago for is to build tools and analyze available high throughput data in order to understand how to predict better combinations of genetic interventions, and also how to integrate all this data together in order to better understand aging. And the tools that my lab is using are tools of bioinformatics, systems biology, machine learning, and we also do some evaluations of the results in vivo, namely in C. elegans. So regarding available data, there are a lot of technologies that have been employed in the last 1 or 2 decades. There is data from different levels of cellular organization. So you have genomics data, you have transcriptomics, you have methylation data, you have metabolomics, proteomics, and so on. The project focused mainly on transcriptomics and methylation data. We looked at the available data sets obtained with microarrays or sequencing, NGS sequencing technology. In terms of metabolomics, there were less resources, so at some point we actually started developing our own database, and Andrei will tell you a bit about it actually in the first presentation. In terms of models of aging, there are also heterogeneous data. So you have the classical old versus young models or experimental designs. You have data from long or short-lived mutants, which you can compare with the wild type. And this both for genetic mutants and non-genetic mutants. Then you have from genomics, you have studies, GWAS studies, and also information on SNPs. This is a bit harder to integrate with the rest of the data, but you can at least inform the results obtained from other analyses. And of course, you can compare genomics or transcriptomics between different species. So after the first coffee break, Anton, Vlad, and Rodrigo will tell you a bit about a study in which we compared transcriptomes of many species. The good news is that data is growing, and probably at an exponential rate. And this is why we try to focus on tools rather than on a specific analysis or on generating data ourselves. So data will become more and more available. We are optimistic. So in the project, we tried more to develop tools and to apply them afterwards. So at a glance, the project was about part of the team looking at data sets, curating them, annotating them, perhaps participating in the development of databases that would help us understand the data or validate the data at the end. We had two colleagues that worked on a portal, a data portal, to host all these data sets and to integrate it with the tools that we develop. We also had to develop some tools to analyze the data sets in a standardized way, because as I mentioned, data is very heterogeneous, and we had many data sets from different labs, different technologies, and we actually had to tweak a bit all the processing, primary processing bioinformatic pipelines in order to make things work smoothly. And then during our analysis, we try to integrate the results from the raw analysis with additional information, from functional information to information about interactions between genes, protein-protein interactions, co-expression data sets, of course, the information known from longevity related databases, the data that we collected for synergism and antagonism. And then we applied methods of bioinformatics, machine learning, pattern searching, and statistical analysis in order to do two main things: to predict combined interventions in C. elegans, and Vlad will tell you a bit about a part of the things that we did to help this inform the sorting of the huge space of combinatorial interventions. And then in the afternoon, Ioan will tell you about one of the combinations that we found to be really successful, extending lifespan more than the single interventions do by themselves. So this was one thing, prediction of combined genetic interventions. And then the other part was to use all this data that we analyzed and that we collected and analyzed to draw some conclusions about aging. And in the second part of the day, you will see some of our collaborations and some of the results that we found related in general to aging. In a more managerial way, so the project was organized in work packages. We had one work package that was focused on dataset collection, annotation, and the raw analysis pipelines. We had work package 2, which was more about research, and this was on how to model the information that we have in a systems biology way to better understand it at the process level, also to compare and do the analysis between species, and generally all the aging related analyses were in this work package. Then work package 3 was more about validation. So we had the machine learning part in this work package, and the prediction of gene combinations and in vivo experiments. And lastly, work package 4 was again a more technical work package, which is development of web tools, hosting the data sets, integrating with the pipelines, building tools, user interfaces for us to understand and work with the data, and also for the creation of databases to show our results. So the first two databases, the web platforms for them were developed by a colleague in this work package. So today we will talk about some of these things. We will talk more about things from work package 2 and work package 3, because these are the more scientific topics. In the first two lectures, we will talk a bit about the data collected and organized in databases related to work package 1. I do hope we will have a very nice workshop, very interesting. And I will stop here maybe for one or two questions, especially organizatorically, if you want to know anything. And then we will move to the first presentation, where Andre will tell us a bit about the MetaBoH database that we nicely collaborated on recently.